Any living organism has to delineate the border between itself and the environment. Periodontal health is a continuing struggle to maintain an equilibrium between microbial attack and defense. Our body is completely protected by an ectodermal sheath that is interrupted only in one site, at the tooth. Special mechanisms were developed in the course of evolution that protect the organism against intruders along the tooth. Exfoliation of junctional epithelial cells and a continuous flow of crevicular fluid removes contaminated cells and bacteria from the sulcus. Relevant components of the immune response are located in the gingiva. Something has changed. The gingival tissue is now swollen and red. Even the most careful probing causes bleeding. Different salivary proteins, including staphyrin and mucins, form a layer on the tooth surface. This pellicle facilitates the initial adhesion of bacteria. Actinomyces and streptococci are among the first colonizers. The bacteria form a sticky polysaccharide matrix that protects them from detachment and creates a distinct environment in which they proliferate and interact. The resulting differentiated three-dimensional biofilm contains interconnecting channels and protects the bacteria from salivary components and antibacterial substances. In the biofilm, bacteria compete for nutrients, exchange genes and signals, and proliferate. Not unlike buds, vacuoles eventually open and release their contents. In the deeper layers, the bacterial flora shifts towards anaerobic, gram-negative, and pathogenic strains. The plaque prevents the inflow of the pellicle proteins that normally inhibit the crystallization of salivary mineral salts by keeping them in solution. Thus, calculus is formed. The subgingival plaque grows in the depth of the sulcus. From here, bacteria and their products, such as lipopolysaccharides and toxins, invade the adjacent tissues. But the body reacts. The innate immune system recognizes bacterial antigens and substances like formylmethionine peptides as foreign intruders and initiates a spontaneous defense. The complement system is activated. A component of this system, the opsonizing factor C3b, labels bacteria for facilitated phagocytosis. Complement factor C5b initiates the membrane attack complex that causes the bacterial cells to burst by forming pores. Defensins, which are ring-shaped host-derived proteins, operate in a similar manner. Meanwhile, bacterial lipopolysaccharides bind to lipopolysaccharide binding protein and are then recognized by toll-like receptor 4. In addition, endotoxins activate the complement system through the alternative pathway. C3a and C5a act on macrophages and neutrophils, stimulating chemotaxis and triggering the release of pro-inflammatory cytokines. Messenger molecules called chemokines cause the endothelial cells to loosen their intercellular contacts and to express adhesive proteins on the inner walls of the blood vessels. As a result, neutrophils adhere and migrate through gaps between the endothelial cells into the surrounding tissue. This process is called diapedesis. They follow the concentration gradient of chemokines such as interleukin 8. This chemokine, also known as CXCL8, is released by the epithelial cells and fibroblasts of the junctional epithelium. Once at the site of infection, the neutrophils kill the invading pathogens after phagocytosis and by releasing reactive oxygen and chlorine species. During the inflammatory reaction, the gingival connective tissue is loosened by shifting the balance between connective tissue protein synthesis and degradation. The responsible collagen-degrading enzymes, such as cathepsin G, are derived from decaying granulocytes, or, like matrix metalloproteinase 8, from macrophages or fibroblasts. The loss of structural integrity of the connective tissues, the dilatation of the vessels, the increased capillary permeability, and the ulceration of the sulcular epithelium contribute to bleeding upon probing. If a new equilibrium between microbial attack and immune response is achieved, the gingival inflammation will persist as an established lesion. Otherwise, an advanced lesion will develop, accompanied by further tissue breakdown and loss of attachment. Bone resorption follows loss of periodontal attachment and is an element of irreversible periodontal tissue destruction. Under normal circumstances, periodontal bone height remains stable as bone formation and bone degradation balance each other. The bone balance is maintained by coupling of the bone-building cells, the osteoblasts, and the bone-resorbing cells, the osteoclasts. Osteoclast precursor cells are derived from immunogenic monocytes, which migrate into the alveolar bone from surrounding blood vessels along a gradient of interleukin 8 and interleukin 1 beta. Here they penetrate through the layer of bone-lining cells. Bone-lining cells present on their surface membrane-bound rank ligand that activates the rank receptors on the osteoclast's precursors. Under the additional influence of monocyte colony-stimulating factor, they merge into multinucleated osteoclasts. What was originally part of the immune defense has now become a bone-resorbing cell. The bone balance is controlled by the ratio of soluble osteoprotogerin and rank L. Osteoprotogerin masks the rank L and is released by osteoclasts under the control of various messengers, including vitamin D and hormones. Once the osteoclasts have completed their task, they attract neighboring osteoblasts by the ephrin receptor ligand system and bone morphogenetic proteins in order to refill the resorption lacuna. The adaptive immune system is involved in the transition from gingivitis to periodontitis by interfering with the bone balance. Naive T lymphocytes become antigen-specific by contact with antigen-presenting intra- and subepithelial dendritic cells. After repeated antigen exposure, antigen-specific T helper cells, the Th1 lymphocytes, synthesize, amongst others, large amounts of new soluble rank L. This rank L leads to osteoclast differentiation. This is mediated by interleukin 17 from Th17 cells. Another T helper cell subtype, the Th2 cells, produce interleukin 4, 5, and 10, causing the stimulation of B lymphocytes, which transform into plasma cells and release immunoglobulins. Osteoclast precursors, being originally immune cells, carry the osteoclast-associated receptor, which can be activated by immunoglobulins. Under continuing presence of bacterial antigens, osteoclasts will amplify bone resorption through the synthesis of chemokines such as monocyte chemoattractant protein 1, RANTES, and interleukin 1 beta. The viable biofilm is removed by supra- and subgingival root instrumentation, which can be accompanied by antiseptics, local and systemic chemotherapeutics. As a result, bacterial load is reduced and the equilibrium is shifted towards healing. The inflammation is switched off in a highly regulated cellular communication process, in which tissue inhibitors of metalloproteinases, lipoxins, resolvins, and cytokine inhibitors are released. Fibroblast growth factor stimulates fibroblasts to crawl into the lesion and synthesize collagen and other extracellular matrix components. Vascular endothelial growth factor initiates angiogenesis. Where viable cementoblasts are still present on the root surfaces, the periodontal ligament may regenerate and new attachment will be formed. Otherwise, the epithelium will grow in an apical direction and tissue repair will occur. Clinically, inflammatory symptoms like swelling, redness, and bleeding upon probing disappear. The firm structure of the gingival tissue is regained. In this way, a new, healthy equilibrium is restored.